Hello, everybody. Thank you very much for joining us this afternoon. My name is Laura Ashram. I'm the Chief Executive at the British Neuroscience Association, and it's my pleasure to welcome you all to this special webinar, which is hosted in partnership with our supporters, Miltenyi Biotec. Today, I'm joined by Caitlin Martin, Product Manager at Miltenyi Biotec, and Dr Jill Fowler, a Senior Lecturer at Edinburgh University, who will be presenting on the topic of global disease-associated glial changes, chronically after focal stroke. So we'd love for you to get involved in the conversation today. Do say hi in the chat and let us know where you're calling in from. And we'll have time for a few questions at the end. So use the Q&A function to submit your questions at any time throughout the presentation, and we'll come back to them towards the end. I'd also like to take this opportunity to remind you that this webinar is being recorded with the permission of our speakers and will be available to watch on demand on the Miltenyi Biotec website. So without further ado, I'll now hand over to Caitlin to introduce our discussion topic today. Thank you, Caitlin.
Thank you very much, Laura, and thank you to the BNE for allowing us to co-host this webinar with them today as part of our supporters package. We're really excited to be here. We're even more excited to introduce our speaker, Dr. Jill Fowler. Dr. Jill Fowler is a Senior Lecturer at the University of Edinburgh and a former Alzheimer's Society Junior Fellow and Alzheimer's Research UK Senior Fellow. Her research focuses on identifying disease mechanisms as a basis for treatments in cerebrovascular disease, brain injury, and dementia, using preclinical models in parallel with studies in human post-mortem tissue. She has developed mouse models to study the long-term consequences of stroke on pathology and cognition, and is currently studying glial cell changes with methods including single-cell sequencing. So without further ado, I'll hand over to yourself, Jill. Thank you very much.
That's great. So thank you, Caitlin. I'm just going to share my screen. Can you see that okay?
Yep.
Yep. And can you see that pointer okay as well?
Yes.
Yep. Okay, so thank you, Caitlin, for inviting me to give this seminar, and thank you everyone for joining online. I'm really excited to tell you about some of the work that's been carried out in our lab. So I'm interested in how stroke can lead to cognitive decline and dementia, and this is very important because the brain is highly vascularized. It's requiring a constant supply of oxygen and glucose to maintain its normal function. So in terms of the brain's voracious appetite, it's only 2% of our body's weight, but it can consume between 20 and 25% of all the oxygen and glucose in our body. And unlike other organs in the body, there's no storage capacity for oxygen or glucose. So the brain is very dependent on this extensive network of blood vessels. So extensive that if we laid them out in a single line, there would be over 400 miles of blood vessels. So any interruptions to the blood supply can have catastrophic effects. And this happens during a stroke. So a stroke is like a brain attack. It happens when blood flow to the brain is stopped, and it can cause a sudden onset of focal neurological symptoms, such as weakness down one side of the body and paralysis. And most strokes are caused by ischemic stroke. So this is where a blood vessel supplying blood to an area of the brain is blocked by a blood clot. And this could, of course, be formed by a thrombus, which is something that occurs locally within the brain, or an embolus, a blood clot formed peripherally that travels into the brain. And this causes a marked reduction in blood supply to the brain and a focal area of brain cell damage, which we call an infarct. Hemorrhagic stroke is also a more minor cause of stroke, and this is where an artery that supplies blood to the brain is where it leaks or ruptures. And this is a particularly catastrophic type of stroke with high mortality and poor outcome. So in terms of the incidence and consequences of stroke, each year in the UK, we have over 100,000 people with a first stroke. So stroke is a leading cause of disability and the fourth commonest cause of death. But we do actually have greatly improved treatments for the acute management of stroke. So we have treatments such as mechanical thrombectomy and clot busting drugs. So this means that we actually have more people surviving long-term after a stroke, but they are living with the serious long-term consequences of a stroke. So up to 1.3 million people in the UK, and over half of these are dependent on other people for help with their everyday activities. And post-stroke cognitive impairment is a major problem. So this can occur in up to 30 to 70% of individuals who've had a stroke. And stroke can also double your risk of developing dementia due to both Alzheimer's disease and vascular dementia. And so the trajectory of cognitive impairment after stroke, you can have immediate or delayed cognitive impairments. And this is dependent on the interplay of a number of modifiable and non-modifiable risk factors and interactions with other pathologies in the brain. So I'm going to introduce this later on in the talk. And so for a long time, many researchers, including myself in my early postdoctoral work, we were very much focused on just trying to understand what happens to neurons after a stroke and trying to develop new neuroprotective strategies. So there was a very neurocentric focus. But then the concept of the neurovascular unit or the neurogliovascular unit came into play to emphasize this unique relationship, structural and functional, that there is between the brain cells and the cerebral vasculature, as well as the glial cells as well. So all the cells in the neurogliovascular unit signal to one another as part of their normal function to provide normal homeostasis to the brain and therefore normal function of the brain. So for example, they signal to one another to control cerebral blood flow, maintain the blood-brain barrier, provide trophic support to brain cells. So I'm interested in long-term pathological changes after an ischemic stroke and the mechanistic link with cognitive impairment and the impact on the neurogliovascular unit. So to understand more, in my lab, I've developed two mouse models of stroke. And the first of these I'm going to talk about mainly today. And this is where we permanently occlude a distal portion of the middle cerebral artery via electrocoagulation. And if we visualize the extent of blood flow reduction here, you can see the MCA branches visualized with laser speckle imaging. So red and yellow are higher areas of cerebral blood flow, whereas blue is low. And we can see that one day after this procedure, we get a marked hypoperfusion in the brain. And then in terms of the pathology, we can see that there is mainly a cortical infarct here, and by 1 month, this atrophies away. So we get considerable atrophy and loss of neurons in the cortex. And in this diagram here, you can see the rostrocaudal extent of the damage to the cortex. So this is the primary infarct. But then in my lab, we were interested in trying to understand what happens to glial cells, specifically microglia, macrophage, and astrocytes. And so we stained sections throughout the brain for IBA1 to look at, first of all, at the microglial macrophage response. And interestingly, we found that this was not confined to the lesion and the peri-infarct lesion areas, which you can see here on this slide scanner image of IBA1 staining. And this is from a section from an animal that survived 1 month after the cortical stroke. But in fact, you can see that the focal area where we get lots of microglial reactivity 1 month post-stroke is actually in the thalamus, and this is an area anatomically connected with the stroke lesion in the cortex. So here you can see the temporal evolution of these changes, some of the morphological changes to the reactive microglia, which by 1 month appear to have an amoeboid morphology. And it does appear like there are an expansion of this population here as well. And if we measure fairly crudely just the percent density of the IB1 staining, we can see that this progresses, and by 1 month we see a marked change here in thalamus. And in fact, it wasn't just within the thalamus that we saw these microglial changes. We saw them spread throughout the brain to other anatomical areas, which were usually anatomically connected with the stroke lesion. So for example, we could see the changes in contralateral white matter tracts, contralateral cortex, ipsilateral thalamus, and internal capsule. And then we also stain sections for GFAP, which is a marker of reactive astrocytes. And this just tells us that there are morphological, transcriptome, and functional changes to the astrocytes induced by the disease process, which in this case is the stroke. So again, here we can see a slide scanner image from a section which is from 1 month post-stroke. And you can see reactive astrocyte changes here in the cortex, but actually more markedly in the anatomically connected thalamus at 1 month post-stroke. And again, looking at the temporal evolution of these changes, we can see that these start in earnest at 3 days post-stroke, but by 1 month, what we're seeing are marked changes to the astrocytes here. So they kind of look like a glial scar, and we can't really see individual astrocytes anymore. And again, like the microglial macrophage changes that we mapped out, we also see these in other anatomical areas of the brain, such as contralateral, white matter tracts, underlying striatum, and so on. So we next wanted to understand more about changes in the thalamus and changes to the inflammatory milieu. So in this experiment here, we have dissected out the thalamus either 1 day or 1 month after the cortical stroke. And we've undertaken some qPCR experiments to look at the inflammatory environment. And supporting our immunohistochemical experiments, we can see the genes encoding GFAP and IB1 are not altered 1 day after the stroke, but they're markedly elevated 1 month post-stroke. And then if we look at other inflammatory related molecules, for example, complement component C3, LCN2, which is a molecule which is upregulated in reactive astrocytes, again, we see these are unaltered at 1 day, but by 1 month, they're markedly upregulated. So as we started to become interested in these changes we were seeing remotely after a stroke, a review was published in Lancet Neurology a couple of years ago, which brought together a number of different lines of evidence and called these changes global brain inflammation. So they brought together clinical imaging studies, such as the PET imaging study I've highlighted in this slide here. So here we can see images from an individual 28 days after a stroke. So they've had an infarct in the cortex. And this is in this study, they've undertaken PET imaging with a ligand which detects inflammatory changes. And you can see that there are not very marked in the peri-infarct lesion itself, but they're actually most marked in the anatomically connected ipsilateral thalamus. So in this review, they were really highlighting that this is an emerging field. We don't know so much about these changes remotely post-stroke, but that they may continuously evolve pathology networks that are required for cognition, and they may affect a patient's long-term neurological outcome. So thinking about what is causing these remote changes to inflammatory glial cells and these changes to the inflammatory milieu, we think that these are caused by a couple of slowly evolving pathological changes. So in keeping with other publications, we found evidence for secondary neurodegeneration in the thalamus. So here we can see these images from 1 month post-stroke. So we can see that there's a loss of NUN immunostaining. So this is a marker of neuronal nuclei, and we can see that there's a loss of them, and the ones that are still there are shrunken. And we've also looked with crescent violet as well, where we can see marked alterations to the neurons, indicating neurodegeneration. And we think in our hands in this model that it starts about 3 days post-stroke, and this would agree with other studies in the literature. So why we're really interested in this is that we think the gradual and delayed manner of the secondary neurodegeneration might provide us with a greater window of opportunity for therapeutic intervention to try and prevent these slowly evolving changes, which we think may be linked with cognitive decline. Similarly, so where we see changes to reactive astrocytes and microglia in contralateral hemisphere is often in white matter tracts. And we think these changes are associated with alterations to white matter integrity. So here we can see sections from MAG immunostaining. So this is myelin-associated glycoprotein. This is a marker that we commonly use in models where there's reductions in blood flow. We see it's quite sensitive to these changes. So here we can see what we call MAG accumulation. So it's accumulations of myelin debris, which were really, extending right into the contralateral corpus callosum and external capsule. So they extend really quite far away from where the primary lesion is. And again, we've quantified this by trying to build a global myelin damage score by assessing damage to the white matter tracts in multiple different anatomical areas. So we can see that this is slowly evolving and likely due to the process of Wallerian degeneration. So we think that this model is clinically relevant because there are clinical evidence of these two slower evolving pathologies after stroke, and they may be linked with cognitive decline as well. So in this study here, we can see an individual with an MCA infarct, and then what happens is there's no changes in thalamus up to 3 days post-stroke, but by 1 year post-stroke, they see that the individual has thalamic iron accumulations, which in the paper they suggest are a marker of secondary neurodegeneration. And they imaged a number of individuals and suggested that these changes in thalamus are independently associated with cognitive outcome. Similarly, other imaging studies with diffusion tensor imaging have shown that there can be altered white matter integrity, which spreads to the contralesional hemisphere and can be associated with poor cognition in the months after a stroke. Individuals who don't have these changes in contralesional hemispheres tended to have better cognition in the period after the stroke. So thinking back to the data where I showed that there was elevated inflammatory markers in the thalamus 1 month post-stroke, there is a knowledge gap about the mechanisms that impact the brain chronically. And one question that we've addressed is which cell type is responsible for these altered inflammatory molecules in this area. So for this, we turned to single cell RNA sequencing. We were interested to understand more about the cell type responsible for these alterations to inflammatory molecules, but just in general about transcriptome alterations in single cell profiles. With single cell sequencing, we can examine cellular heterogeneity post-stroke, and what we hope to move towards doing is determining ligand receptor interactions between glial cells. So we're specifically interested in astrocyte microglial interactions, and we can try and tease this out with programs such as CellChat, where we can look at ligand receptor alterations between our interested cellular populations. So in this specific experiment, I undertook stroke in mice that were aged 5 months, and these mice survived for a further 1 month. And then I dissected out the thalamus so we could look at the grey matter changes associated with secondary neurodegeneration, and also the corpus callosum to look at white matter tract changes. We dissociated the cells with the Miltenyi adult brain dissociation kit and used a panel of their antibodies, which allowed us to extract the key glial cell populations, astrocytes, oligos, microglia, and endothelia. And this experiment was undertaken in collaboration with Dr. Andrea Corsinotti at Edinburgh University, who runs a single cell omics facility, and has really been so helpful in helping us develop these technologies. And so these data were analysed under the supervision of Owen Dando from UK DRI here at Edinburgh, and really accelerated forward with a talented MSc student I had in the lab last year, Erin Reardon. And so the general workflow she was following is shown here. So the data is analysed in RStudio, and we undertake a number of quality control steps, such as removing poor quality cells, doublets, and applying a mitochondrial filter. And then we form what we call UMAPs, which I'll show in the next slide, where we cluster together cells which have similar transcriptomes, and we try and work out what cellular populations they are. And then we can go on and undertake our analysis. So we can do differential gene expression, either comparing stroke versus sham, for example, or even comparing different clusters from the same animal. So here we can see the first level cluster analysis that we undertook. We had 17 clusters, and so we can see all the glial cell populations that we expect to see based on our FACS sorting strategy. So we have microglia, astrocytes, and oligodendrocytes. So another way that we can present these data in a UMAP is by colouring the cells pink if they came from sham treated mice, or blue if they came from stroke treated mice. So here you can see one population really stood out to us, which came almost exclusively from the stroke treated mice. And so when we, excuse me, when we determined what cell type this population was, we found that they had this signature of disease-associated microglia, which I'll explain in the next few slides. So Erin went on to subcluster just the microglia macrophage cells. And here we generate a new UMAP, where we have 5 homeostatic microglial populations, but we were interested in these 2 disease-associated microglial populations. And we also had some other immune cells as well. But again, if we color codes these as coming from either thalamus or corpus callosum post-stroke, we can see that the DAM1 population largely comes from the thalamus, whereas the DAM2 population mainly came from the corpus callosum. So this is interesting that there are transcriptome differences between these 2 anatomical areas. The disease-associated microglia, this is a cell state, a signature that was first reported in Alzheimer's disease mice from microglia that surrounds amyloid plaques, and has since been confirmed in AD tissue and other models of disease such as multiple sclerosis. So I'll talk you through these changes, and remember that the disease-associated microglia cluster was at the bottom here. So we can see upregulation of genes involved in lipid metabolism, such as ApoE and LPL. There was an upregulation of cell surface receptors, such as TREM2. Genes involved in lysosomal function, cystatin 7, and genes involved in tissue remodeling, such as SPP1. So SPP1 was actually one of the most significantly upregulated genes. And so in collaboration with Barry McCall's lab, who have great expertise in microglial changes in disease states, and with the help of Jack Barrington, Erin and I undertook some RNA-scope experiments where we could spatially confirm that SPP1 was upregulated, and we could look at its location. And so we did see SPP1 cells in corpus callosum and surrounding the lesion as well. But here you can see this very marked upregulation, this focal area of SPP1 induction in the thalamus. And this is 1 month post-stroke. So that was really interesting to us because other papers suggest that SPP1 knockout mice, although they don't have difference to their primary infarct volume, that they tend to have more secondary neurodegeneration post-stroke. So this perhaps suggests that these SPP1 microglia are performing a protective response in the longer term post-stroke. Excuse me. And so we also undertook differential gene expression analysis. So here we were comparing the homeostatic microglia, whether they came from sham or stroke mice, with the disease-associated microglia. We can see some similarities and some differences when we look at the thalamus versus the corpus callosum. For example, in common with this cell state signature, we see a downregulation of homeostatic genes and receptors such as P2RY12 and TMEM119, whereas we can see the top 10 genes that were induced have some similarities and differences. So SPP1 was the most induced in thalamus, whereas in white matter, we see a significant induction of ApoE, and LPL is in the top 10 as well. So this is perhaps suggesting that there's an ongoing process of myelin repair in the corpus callosum. And if we look at different groups of genes and the pathways that link them together with gene ontology analysis, a number of the different gene pathways that were altered made absolute sense to us. They were involved in immune cell function, cell proliferation, response to cerebral ischemia, innate immune response, for example. But we can see, again, some interesting region-specific alterations. So in thalamus, one of the most upregulated pathways was complement-mediated synapse pruning. So perhaps suggesting that there's ongoing process of removal of these degenerating synapses and maybe some synaptic remodeling. Whereas in corpus callosum, we have this pathway involved in positive regulation of myelination, perhaps suggesting that there's ongoing myelin repair. The one further exciting finding that we had was the presence of disease-associated oligodendrocytes. So a couple of years ago, Ido Amit's group identified an oligodendrocyte state in Alzheimer's disease mouse models and in AD tissue that was associated with brain pathology and correlated with cognitive impairment. And they termed these disease-associated oligodendrocytes. So again, when we subcluster just markers of oligodendrocytes and then color code them as to whether they came from sham or stroke, we can see this blue population, which comes almost exclusively from the stroke mice. And then we searched for genes that were upregulated in this population or altered in this population, and we saw, for example, 2 of these disease-associated oligodendrocyte signatures were induced. So one of these is the protease inhibitor, SERPINA3N, as well as the inflammatory molecule complement component C4b. So I think this is the first time that changes in oligodendrocytes have been reported chronically post-stroke, and we're interested in this and trying to find out more about pathological changes to oligodendrocytes in our model. Complementing this work with some single nuclear sequencing from human post-stroke tissue. So this is a collaboration with Professor Raj Kalaria from Newcastle University, and we've undertaken single nuclear RNA-seq on individuals with a history of cortical stroke. So these were individuals that were included in the COG-FAST cohort. So this is a longitudinal study of dementia incidence following stroke in the over 75s. So here we can now compare individuals who've had a stroke who went on to develop dementia with those who had a stroke and did not develop dementia. So we can try to tease out transcriptome alterations that confer resilience to cognitive decline post-stroke. So here we sequenced 15 libraries. Again, this was in collaboration with Andrea Corsinotti, who helped build the libraries, but also in collaboration with Professor Anna Williams and Louise Seeker, a postdoc at that time in Anna Williams' group, who've developed a number of the techniques that we used in human tissue. So the tissue we obtained was from thalamus, from cases that Raj had identified as having a history of cortical stroke. And then from 20 micron cryosections, we isolate the nuclei with nuclear isolation protocols, and then we go on to sequence them. Again, Erin undertook some of the workflow for this experiment. So again, similar workflow as for the single cell experiment in mice. So we ended up with 25,000 cells in this case. There was a bit more heterogeneity in terms of the quality of the libraries, which is something we would expect from a human post-mortem study. So we did have 4 cases where we saw not such a great quality of the libraries, but luckily these were evenly distributed amongst the 3 groups that we were studying. So first, look at our UMAP from the human study. We can see again all the glial cell populations that we're interested in, as well as neurons. So with human post-mortem tissue, what we're doing is sequencing the nuclei, which are relatively well preserved. So we can't undertake single cell sequencing and look at transcripts in the whole cell. We're just looking in the nucleus. But this is not using any sort of cell-specific approach. So this was just unbiased. And so now we also have the presence of neurons and neuronal precursor cells as well. So based on the fact that we saw really interesting microglial changes in mouse, we wanted to see if we saw them in human as well. So under the guidance of Jack and Barry, we subclustered the microglia and immune cell populations. So we can see here now that we have 4 microglial populations, as well as a number of other immune cell clusters. So the key problem that we've had with this study is that we have relatively low numbers of microglia that were sequenced. There may be superior methods to try and sort more specifically for microglia that we're trying to develop just now. But for this study, even when we split our UMAPs into the 3 groups that we were interested in comparing, so the control, stroke with dementia and stroke without dementia, even though we're limited in number, we can still see some interesting findings. So we do see fairly distinct transcriptome profiles. For example, in control, almost exclusively only in the controls, we have this human leukocyte interferon cluster, which contains a number of genes involved in interferon signaling, immune response, and the reactive microglial state. So perhaps suggesting that this is conferring resilience to developing dementia. And again, in the stroke without dementia group, we have this enriched CD163 population. So this CD163 expression has been associated with anti-inflammatory properties, increased phagocytosis, and has been proposed to have a role in preventing cognitive decline. So that was really interesting. Because of the low number of microglia we sequenced, we don't see many genes changed when we look with differential gene expression analysis. But of the few that were altered, they were still really interesting to us. So for example, this one gene called CEBPA, a transcription factor, which is upregulated in stroke without dementia. So this other papers have described this as being upregulated after cerebral ischemia. And an in vitro study in Glial from a few years ago also suggested that this may be a transcription factor involved in the conversion into a dam-like state and may be involved with phagocytosis. So this may be involved in conferring resilience to developing cognitive decline post-stroke. So in summary, we've shown that stroke causes a focal area of brain cell damage in the short term. So we see in the mouse model that we see a cortical infarct. But weeks and months after the stroke, there are widespread inflammatory cell changes in the brain, for example, in thalamus and contralateral corpus callosum external capsule, where we find disease-associated microglia and oligodendrocytes. So we're interested in understanding more about these really chronic changes to try to understand if we can target them to better rewire the brain after a stroke to prevent cognitive decline. So for this, we have a number of other areas of research ongoing in the lab. For example, we've undertaken some studies where we boosted astrocytic NRF2, which is an antioxidant factor. And we're also trying to develop some chemogenetic approaches to upregulate a protective pathway in astrocytes, and that's a collaboration with Giles Hardingham from DRI. So our next steps are confirming these gene changes with RNA-scope, as we've done with SPP1, and looking for protein changes with immunohistochemistry. We really want to try to understand what's actually causing these glial cell changes, what's the underlying mechanism, for example, what drives the secondary neurodegeneration in thalamus. There'll be a loss of trophic support from the cortex from those cells that have died, and what's causing the white matter damage as well. And then we're interested in how risk factors that are associated with poor cognition after a stroke, how these impact these temporal and spatial alterations. So I'm going to speak briefly in the rest of the talk about aging and our efforts to model concurrent amyloid and ischemic stroke. And we're also interested in, as I said, in how cell-to-cell signaling is altered after a stroke. So we're trying to develop bioinformatic approaches to look at ligand receptor interactions. But another way we can do this So one cell-to-cell interaction we're interested in is astrocyte microglia interactions. So we can do this by using a line of mice that are deficient in microglia. So when there are no signals from the reactive microglia, what happens to the astrocytes and to the other ongoing pathologies? So for this experiment, we use fire mice. So these are deficient in an FMS intronic regulatory element, which is necessary for the normal expression of the CSF1 receptor, which microglia need for their normal function and proliferation and so on. So fire mice are born without microglia. They develop a normal central nervous system. Changes to other neurogliovascular cells are relatively unaltered. So this is a good approach for us to understand what happens to astrocytes and what happens to other pathological features in the absence of microglia. So we've undertaken a small cohort pilot experiment, and this was with Cornea Pretorius, a master's student in my lab, and we have undertaken stroke in fire mice or wild-type littermates, and this model was developed by my collaborator at Edinburgh, Claire Predans. So first of all, we determined if there were any reductions in alterations in cerebral blood flow in these mice compared to their wild-type littermates, and we can see marked reductions in cerebral blood flow at 1 day, sustained by 3 days, but these didn't seem to be altered when we compared the fire mice with the wild-type mice. So then we've looked at the primary infarct volume, so looking at the neuronal damage caused by the stroke. And we can see here when we compare wild-type and the fire knockout mice that there was no difference. There was no statistically significant difference in the wild-type versus the fire knockout mice. So that's interesting in itself, that without microglia, the lesion is not particularly enlarged. And this allows us then to go on and study what happens to glial cell populations and other pathologies remotely, knowing that there's no alteration in the primary infarct volume. So for example, here we can see in thalamus in the wild-type mice, 3 days post-stroke, which is the start of these changes in thalamus. We can see that there's an induction of reactive microglial alterations, whereas obviously there's none in the CSF in the fire knockout mice. So one interesting preliminary finding that we see in this model, so as I mentioned earlier in the talk, we start to see secondary neurodegeneration about 3 days post-stroke. So Cornea stained her thalamic sections with cresol violet, and she formed a scoring system depending on the extent of the neuronal degeneration. So 0 was no abnormal cells, whereas 2 had the most abnormal cells. And interestingly, she graded, obviously in a blinded way, but it turned out that most of the mice with score 2, which had most secondary neurodegeneration, actually came in the fire knockout mice. So this perhaps suggests that these microglial changes that we're seeing in thalamus are part of a protective response, and when we don't have them there, we're maybe seeing more secondary neurodegeneration. But we're interested to find out more about this, and we want to try and do longer term studies. So I'm also interested in other risk factors. So for example, aging is the biggest non-modifiable risk factor for the development of post-stroke cognitive impairments, and many functions in the brain, such as reductions in cerebral blood flow, increased oxidative stress, many of these are altered with aging. We've undertaken one study where we compared and contrasted stroke in either 3-month-old mice or 18-month-old mice. And these mice had a battery of cognitive tests, and then they were culled 6 weeks post-stroke. So we're characterising the pathological changes to these at the moment. And this was carried out by master student Ellen Gray in my lab, also very talented, who worked really hard on this study. And she found that there was a deficit in longer-term spatial memory. So this is a task where we block off one arm of the Y maze, and this is assessing spatial working memory. So if we leave the 1 minute intertrial interval to look at very short-term memory, we see that it's unaltered. But when we look at longer term inter-trial intervals up to 30 minutes, we can start to see that there's an effect of stroke when we do our two-way analysis, and that this is exacerbated by aging. And also Ellen looked with novel object recognition. So this is just a form of memory, which basically is exploiting the fact that the mouse should spend more time exploring a novel object. And so this is assessed by something called the discrimination index, which is just a measure of how long the mouse spends exploring a novel object. And this seemed to be impaired in the aged mice post-stroke. So the ongoing work, we're looking at the correlates of these cognitive changes with pathology. We have undertaken a small cohort just in the thalamus, where we're looking at single cell sequencing changes to the astrocytes and microglia. We're looking at cellular proliferation and just asking if secondary neurodegeneration and white matter integrity alterations are exacerbated with aging. And finally, another study we have ongoing in the lab is just looking at interactions between amyloid and stroke pathology. So we're modelling this in Swedish Dutch Iowa mice, which have both vascular and parenchymal amyloid accumulations. And interestingly, again, we don't see any exacerbation of the primary stroke lesion. And here we actually looked at synapse loss as well by crossing these mice with a line of mice that had a fluorescent tag on excitatory synapses. So there's a loss of synapses in the stroke infarct, but it's not exacerbated in the Swedish Dutch Iowa mice. So there's no interaction of the stroke pathology with the amyloid pathology to cause changes to synapse loss, infarct volume, or glial cell changes in the primary infarct. But interestingly, what we do start to see is that the secondary connected regions, such as thalamus and internal capsule, we see that there is a genotype effect now. So we see more reactive astrocytes and microglia, for example, in thalamus in this model, which was actually a model of subcortical stroke. So lastly, I just want to quickly tell you about a really exciting grant that we've had funded. So we're really privileged to have been awarded this 5-year grant from NIH in the States. This is a 5-year program where we're investigating perturbed microglial endothelial interactions as a mechanism underlying white matter abnormalities, which may drive cognitive decline in vascular cognitive impairment and dementia. So this is a program which is led by Professor Karen Horsburgh, and also including myself and Barry McCall at Edinburgh and Axel Montaigne, as well as Professor Raj Kalaria from Newcastle University. So this work will involve continuing on some of the single nuclear sequencing from the CogFast cohort. And with Catherine Hall and Yuri Kudelka, they'll be using some really cool multiphoton imaging to image microglia longitudinally in white matter. So we're using a number of really cutting edge techniques, including some spatial and the single cell nuclear techniques I've been describing, as well as cognition using a novel PET ligand. So I just wanted to flag that there'll be lots of exciting opportunities on this project. So we're looking for masters, PhD students, and an in vivo postdoc, as well as an in vivo, as well as a postdoc, who will help undertake the bioinformatic analysis. So please do reach out to any of us if you want to discuss some opportunities on that project. So all that would remain for me is to thank everyone in my lab, past and present. I tried to highlight everyone as I went along for their really hard work in bringing together these studies. Undertaking long-term studies in stroke is not an easy task. So I'm very grateful to everyone in the lab who worked so hard on this day in, day out. And I'm also really grateful to our funders, so AR UK, as well as smaller pots of money which have been so helpful for us. For example, the RS McDonald Charitable Trust, Wellcome Trust. And I'd like to thank everyone from Edinburgh Neuroscience who's helped contribute towards these studies. So do you have any questions, please? I'm very happy to answer questions just now or later on via email. And please also reach out to us for any to discuss any opportunities that may be available on the NIH grant as well. So thank you very much for your time, everyone.
Fantastic, thank you so much. That sounds like a fantastic opportunity as well. I know there's obviously the NE Careers Fair coming up in the next couple of days, so it's certainly something that'll be on everybody's mind. So there are a couple of questions in the Q&A function, so we'll try and get through, I guess, as many as we can, and any that we can't get to, we'll try and answer separately. So we'll just start off with the most recent one. So have you induced stroke in other areas of the brain, and is the glial response similar irrespective of location?
Yeah, so that's a really great question. Thank you. So I think I briefly alluded there to the fact that we've also undertaken some studies with a different model of stroke, which is intraluminal thread. So normally we would have used this model with 60 minutes of occlusion and it causes a very large infarct. But when we reduce that down to 15 minutes of occlusion, we see damage mainly in the striatum. And so what we'll start to see is glial cell changes in anatomical areas that are connected with the striatum. So globus pallidus, internal capsule, substantia nigra. We're tracking right back to these regions, cortex and white matter tracts as well. So it is different, but it's dependent on the fact that it's a different area of the middle cerebral artery territory that we've damaged. Yeah, thank you.
Fantastic. And another one we've got, so just starting off, just to say thank you for the talk, but they're asking what would happen to the inflammation profiles if there's recurrent stroke, and could we predict the inflammation profiles would plateau at any point of infarction?
Yeah, that's a great question. Yeah, thank you very much. So obviously recurrent stroke is a big problem. So individuals having had a stroke is one of the most common causes of recurring stroke. So if you've had a stroke, you're more likely to have another stroke. And so that's something we're really interested in trying to develop a model for. Even we're just interested in trying to induce aging-like changes in the brain with a hypoperfusion and then inducing a stroke to see how that will alter the inflammatory molecules. I don't know what would happen, to be honest. It would be so interesting, wouldn't it, if the changes that we see initially after the first stroke are like a glial priming sort of mechanism, and that then alters their subsequent susceptibility to inflammatory changes, which may be more marked or perhaps even protected against because of some of the transcriptome changes that we see caused by the first stroke. So that's really interesting. Yeah, thank you. Something to think about.
And I've got another question here, is kind of more around the procedure. So just asking around if you can briefly outline the procedure used for inducing stroke in the animal models, and in particular, how long was the blood flow blocked to the animal models?
So for the study that I spent most of the time discussing in the webinar, so this is actually a permanent occlusion of a distal portion of the middle cerebral artery. So what I've done is drilled a hole in the skull where we can access and see the distal portion of the middle cerebral artery, which supplies blood flow to the cortex. So we use an electrocoagulation device, and that should permanently occlude that vessel. So that's leading to permanent infarct there. So that's how that model works. And then I was also briefly alluding to using an intraluminal thread model, and that's introduced into the common carotid artery, and it occludes the origin of the middle cerebral artery, and then that's withdrawn to allow for reperfusion after a certain period of time. So it's a modest duration of blood flow reduction I've been using, which is 15 minutes, and that causes mainly striatal damage. So there are advantages and disadvantages of both these models. So for example, reperfusion is something that's quite clinically relevant that we'd like to model, whereas the electrocoagulation, we're seeing this pan-necrosis of cells in the cortex as well. Thank you.
Thank you. And there are a couple more, which I think we've got some time for. So are there any significant associations between different types of dementia with the gene expression or glial activity in stroke patients? Sure, that one.
Yeah, just having a think about that. Significant association between. Yeah, so I don't think we've necessarily tried to address that. So most individuals in the CogFast cohort would have had a diagnosis of vascular cognitive impairment, so vascular dementia. So the changes we're seeing there are all we've studied at the moment are the microglial changes. So like Alzheimer's disease, there's, a lot of marked microglial changes which are associated with the cognitive decline. When an individual has a diagnosis of dementia and when they die, we look at their tissue post-mortem, they likely have multiple pathologies which may be features of Alzheimer's disease and vascular dementia as well. So even an individual with Alzheimer's disease likely has lots of vascular damage as well. It's quite a hard thing to tease out.
Yeah, thank you. And we do have a few questions just around the thalamus as well. So do you have any idea at the moment why the thalamus is showing glial changes and not other anatomically associated brain regions?
So we focus in particularly in the thalamus because we're seeing the secondary neurodegeneration, which clinical studies have suggested might be associated with cognitive impairment. And so that's interesting for us to understand mechanistically why we are seeing those changes and if we can try and target them. But we do see changes to other anatomical areas that are connected with the cortex as well. So we're seeing changes in contralateral cortex and the white matter tracts that are connected with it as well. So it isn't just thalamus, it is spreading quite globally, but not in anatomical areas that are not connected with the cortex, if you see what I mean. So it's quite widespread, and that's really interesting to us.
And just another question around the thalamus. Obviously, throughout the talk, you referred to, you heard a lot to the loss of neurons in the thalamus. Which particular part of the thalamus were you kind of referring to? Or it's kind of more globally in the thalamus?
Yes, no, I'm trying to think what they're called. I think I only know them as VPL and VPM. I can't think they're the ventral, posterior, and lateral. There's specific thalamic nuclei. And they are the parts that are connected with the parts of the cortex that we've damaged. So yeah, I think it's something we really need to try and understand more about which specific parts of the cortex were causing damage in and which specific thalamic tracts we are damaging. So it's maybe something we could do tracing studies on to examine more about the specific nuclei that we're damaging.
Thank you. We do have a couple more, and I think we have enough time. I'm gonna go for the next one. So you mentioned that there was a time window for therapy that would impact on the recovery. Would the age of the animals potentially shorten that window?
Potentially, yes. So that's something. So we've only looked in our aged mice, we've only looked 6 weeks post-stroke. So it would be really interesting to conduct some temporal studies where we look earlier, like 3 days and 7 days, to see if it is accelerated with the ongoing processes that we see with aging. So even in our sham treated aged mice, we for sure see marked changes to the thalamus. So we see the occasional really wild looking reactive microglia. We see lots of vascular changes, I think. So we can see microglia around the endothelial cells. So we can see really thickened vessels there as well. So it would be interesting for us to look sort of temporally at shorter term changes in the thalamus and try and see if they are exacerbated with aging processes. Yeah, thank you.
Thank you. So we've got a slightly longer question here. So it is, considering the post-stroke global inflammation, do you think that these insights could be applied to primary neurodegeneration? In Alzheimer's, protein pathology builds up in the brain and individuals function cognitively well for years, but then suddenly decline. Could this sudden cognitive decline possibly be linked to that increased immune activation?
Yeah, so I think there's probably common mechanisms that we're seeing here post-stroke that we see in other neurodegenerative diseases. So we can see for example, in Alzheimer's disease, this sort of very well-defined temporal progression, things starting in entorhinal cortex, spreading to hippocampus, and then, frontal cortex and so on. And it is just like almost like a prion-like spread of degeneration. And I'm sure that the changes must be quite similar. It would be something that would be really interesting to look at. But I think there was a second part to that question. Was it just about the...
It was just around whether the sudden cognitive decline could be linked to the immune activations.
Yes, I guess so. Trying to target that immune activation to see if that can alter cognitive decline would be useful to try and address that question. And it's something we've been trying to do with our mouse models, develop ways of looking at cognition so we can try and answer questions like that. Yeah.
I think we've got time for one or two more. So the next one is, did you look into the difference between microglial profiles in female versus male mice post-stroke? And if you did, kind of what was the findings? Did they respond differently at all?
So no, unfortunately, we've not looked at female mice, and that is absolutely something that we should do, and we should move forward looking at. So very often in the stroke field, we do tend to carry out our studies in male mice, and this is because in female mice, depending on the point they're at in their estrous cycle, if they have high levels of estrogen, that can be neuroprotective, so that can alter the primary infarct. So that's kind of been our justification for not using female mice, but I don't think that is really moving forward. I think we have to include female mice and just understand these changes as well. So we know, for example, in Alzheimer's disease models, that microglia changes are more marked in female mice compared to male mice. So that's absolutely something that we must think about moving forward. Yeah, thank you.
Fantastic. And the next one is just saying it's a great talk and thank you very much. But did you look at the neurotoxicity of the reactive astrocytes in the presence versus absence of microglia at all?
Yeah, so that's something that we'd obviously like to do. So we're trying to get some funding to look at single cell sequencing and single nuclear sequencing of what happens to reactive astrocyte phenotype in fire mice and in their wild type litter mates. So we can try and compare and contrast our astrocyte transcriptome alterations with other changes that have been already published in Alzheimer's disease models, for example. So that's something we'd like to do. I have to say that we've been finding the astrocytes quite tricky to study just in general in our single cell model because they have glial scar-like features. And so I think they're actually quite hard to extract from the brain and have survived through the single cell process. So it could be that spatial transcriptomics, so we're going to undertake a study with cosmics where we can look at spatial changes to reactive astrocytes and hopefully move forward with some single nuclear sequencing as well.
Fantastic. So we've got 1 minute and one more question, so we'll see how we get on. What would happen in the cognitive learning of the CSF1R fire knockout mice post-stroke?
So we've not undertaken that experiment, so I'm not sure what would happen. I think we'd hypothesize that we might see more cognitive decline. I think overall our results are pointing to the microglial changes as being protective. And so I think in the absence of those, we might see more neurotoxic, like we were discussing in the last question, more neurotoxic like astrocyte changes, which might lead to more white matter tract damage and secondary neurodegeneration. So the fire mice study I described was in 3 days post-stroke. So we've not had a chance to look at any longer term studies to look at cognition. We know from the work of our colleague at Edinburgh University, Veronica Miron, that there are very subtle alterations to cognition that we really have to tease out with quite complicated cognitive tests where we look at cognitive flexibility, but there might be subtle changes to myelination with aging and some very subtle changes to cognition. So we would, I guess, hypothesize that with stroke that would be exacerbated, but something we'd be really interested to look at.
Oh, fantastic. Well, thank you very much. We are out of time now. There is one more question, but we can try and get back to that separately. But yes, I guess thank you very much to yourself, Jill, for taking the time to talk us through that presentation, but also answering all the questions, because we've got through 12 questions there, which is quite impressive.
Yeah, no, that's great. Thank you everyone for submitting these questions. It was really great to have.
Thank you very much. And also thank you to the BNE for co-hosting this with us as well today, because we couldn't have done it without you guys.
Yeah, so thank you, Caitlin, for inviting me to give this talk as well. It's been great. Thank you.
Yeah, thank you very much, everyone. Have a nice rest of your day.